Biosensors are analytical devices that combine a biological component with a physiochemical detector. In the context of microbiology, a biosensor is a device that can signal the presence of a disease causing microbe. This can be in a sample such as water or blood serum to help diagnose infections. There are multiple types of biosensor devices, such as enzyme based, tissue based, immunosensors, DNA biosensors, and thermal or piezoelectric biosensors. Today, I focus on DNA based pathogen detecting biosensors, most common of which are lab on a chip type of device that can use microfluidics and sense the change in chemical activity in your sample. We can use the DNA of certain pathogens to flag certain foods as unhygienic, for example, with the detection of bacterial species like E. coli or Staphylococcus aureus. A lab on a chip involves six main parts: the sample injection, followed by a large number of micro pumps or valves on the chip that can precisely control the flow direction and the flow rate. Then, the sample preparation includes the capture of the pathogen using cell lysis or nucleic acid extraction, etc. This part often needs external electric field or a magnetic field or even ultrasonic sound waves. Then there is the nucleic acid amplification. Since we are working with DNA, we can use something in addition to traditional PCR, such as an isothermal nucleic acid amplification, so that we can control the temperature of the reaction process. Finally, there is the detection of certain pathogens using techniques such as fluorescence or electrochemical. Fun fact, I once participated in a project where we built an automated drug dosage monitoring system by using a chemical compound as a biomarker for epileptic seizures. This model had revolved around the idea that fluctuations of certain chemicals in the blood can uniquely be associated with disease onset. And it is true that such devices in the future can aid in more personalized medicine delivery. However, today most traditional detection methods are expensive, time consuming, and unfeasible in practice in the absence of sophisticated instruments or even trained operators. The lab on a chip kind of a biosensor is an emerging point of care technology that has a good potential to integrate a laboratory setup onto a single monolithic device. There is already ongoing research showing that we have detection limits as small as just 10 cells of the pathogenic bacteria, and these can even be detected within the span of an hour of application. At the same time, there is a limitation that certain steps of DNA extraction and purification need to be done as a precursor to the biosensor application. So, although this device is useful in detecting food poisoning or even monitoring the hygiene, these bulky pre processing steps make it unable to meet the requirement for a point of care technology today. This brings us to bioreactors. A bioreactor is a vessel in which a biological reaction or chemical change can take place. The biological systems involved are with enzymes, microorganisms, animal cells, plant cells, and tissues. So, why do we need these large contraptions? Unlike small scale biosensors, a bioreactor can be scaled to large industrial level production of chemical compounds or cultivating tissues or cells or even monitoring chemical reactions. More interestingly, a bioreactor can be optimized to certain growth conditions that are perhaps favorable for certain microbes. We can use the pH, the temperature, nutrients, and the aeration as techniques to do this. Specifically on context with the human gut, we can develop an in vitro gut intestinal system that can be used to study digestion, nutrient absorption, and the gut microbiome without the need for any human or animal trials. In fact, this goes hand in hand with the ethical principles of animal welfare. This was first laid down by William Russell and Rex Burge in 1959. As a side note, these principles are also known as the three R's: reduction, refinement, and replacement, ultimately to promote more humane methods to generate experimental data. A gut bioreactor can be of four main types: A complex multi-compartment simulator which can model multiple regions of the gut, including stomach, small intestine, and the colon, simultaneously to study the entire digestive process, or a specialized colon model which can, for example, focus only on the colon where the majority of microbial activity occurs. They can also be gut on a chip, which involves microfluidic systems to use living human cells and observe any host to microbe interactions. And finally, there could be specialized bioreactors to study species to species interactions or to specifically replicate biomechanical or chemical process in the stomach. I have collected some examples of each of these four bioreactor groups, with the most popular one being a complex compartment machine known as the SHIME system. SHIME stands for the Simulator of Human Intestinal Microbial Ecosystem, and it mimics the entire gastrointestinal tract and allows long-term studies of the microbiota. There are multiple extensions of the SHIME system, such as with the addition of a mucin-covered beads to allow any study of the bacteria that adhere to the intestinal wall. This is known as the mucosal SHIME system. Similarly, there are the toddle SHIME system that is adapted for a toddler's gut, or something that is controlled by a dynamic computer system known as the TIM system. There could be specialized colon models, for example, the polyferm, which stands for polyfermentor intestinal model, and it uses immobilized fecal microbiota in gel beads. This can allow parallel operation of multiple reactors to compare the different treatments on the same microbiota. With the gut on a chip system, and allows high throughput screening of gut microbiota modulation. Finally, there are specialized species simulators, which can be for specific animal gut intestine systems like pigs, dogs, and cats. Another popular bioreactor is the mini colon model, which is an example of a specialized colon model. This is also known as MICOMO and is relatively low cost benchtop device. It makes the research on different gut regions more accessible as it consists of replicate bioreactors that can be run independently. This allows for high throughput multiplexing capability without requiring a fully anaerobic chamber. We also have the RoboGut, which is capable of maintaining precise computerized control of the environment, like the pH, the anaerobiosis, ensuring reproducibility across different experiments. An application of this RoboGut system involves the evaluation of gut microbial community and metabolic functions. Note that this is also a big machine setup and may emulate results closer to an in vivo experiment. Similarly, there is the PETER system for the peristaltic mixed tubular bioreactor, which is designed to mimic the peristalsis movement. This part of digestion in the GI tract is often overlooked. So, PETER features a single tube design that can be periodically mechanically massaged to simulate intestinal peristalsis. Along with an integrated dialysis system, PETER is a very useful and realistic implementation of the GI tract. Ultimately, the choice of experiment decide which biomechanical method best suits a research investigation. Bioreactors can be an all-in-one solution to generate and monitor pathogens in a controlled environment, whereas biosensors can be used to flag the detection or presence of a certain chemical compound. In most advanced systems, biosensors are usually inbuilt with bioreactors to continuously leverage the functionality of monitoring as well as identifying certain bacteria.